This conference will now be recorded.
A very good afternoon, Vignesh. Thank you very much for joining us from Singapore on this online webinar. We have three topics we'd like to get your opinion and thoughts on. And the first of the three topics is, for a typical biopharmaceutical drug substance manufacturing process, what would be the typical unit operations and what would be the sequence?
Okay, the typical unit operations from first to last in this sequence are upstream fermentation and processing, cell breakage and cell disruption, and then followed by downstream processing. The downstream processing stage is divided into two different unit operations: solid liquid resolution, for harvesting and recovery, and chemicals separation and purification. Under harvesting and recovery, it's further subdivided into two unit operations: solid liquid resolution and isolation of a group of compounds with similar physical properties from the rest of the biomass. And following downstream processing, we have finishing of the purified protein bulk, followed by an optional formulation stage and packing and freezing. And to elaborate a bit on the fermentation stage, fermentation involves engineering and growing the cell line under controlled conditions by supplying the cells with various nutrients which they require to grow. And then after a series of biochemical reactions, the bacterial and microbial cells will produce the protein products, will express the protein products of interest. These can be either intracellular or extracellular. In intracellular systems, the protein products are expressed inside the cells, and in extracellular systems, the proteins are the desired protein product, the active pharmaceutical ingredient, is expressed outside of the cells. So for extracellular systems, we don't need to go through an extra step of cell disruption and cell breakage. But for intracellular systems, we have to release the protein product from inside the cell to outside the cell. So we have cell disruption and cell breakage. These are typically three methods: either mechanical grinding, or using detergents and enzymes, and also by high pressure methods such as homogenization and centrifugation. Centrifugation is one of the most commonly used methods for cell disruption and cell breakage. Yeah.
That's good. Thank you very much for that elaboration, Vignesh. That was good. Thank you. That was a good, competent answer. And then a similar question for drug products. So for a typical biopharmaceutical drug product, what would be the sequence of unit operations we would encounter?
Okay, the sequence of unit operations for drug product manufacturing in a typical biopharmaceutical manufacturing facility from first to last are thawing, formulation, sterile filtration, filling, lyophilization, inspection, labeling, packaging, and bioanalysis. So these are the 9 steps. So under the first step of what was that? Let me see. What did I just? Sorry.
Okay, take your time. Take your time. You're okay.
Yeah, under thawing, the required frozen bulk is thawed out to adjust to the ambient temperature. And then the next step is formulation, whereby the thawed protein purified bulk is mixed with additives, inert ingredients, excipients, and other buffers to formulate to the required strength. And it is this formulation step that converts the drug substance into a drug product. And the next step will be sterile filtration, because we have to remove bacteriological contaminants. So we pass it through a 0.22 micron bacteriological sterile filter to remove bacteriological contaminants and also to exchange buffers. And the next step will be filling. For filling, we can either have manual filling or automated filling. For manual filling, we have a operator in the filling room with the filling equipment who transfers the transfer card to the vial depyrogenation stage, where the depyrogenated vials are transferred onto the transfer car. And once this is done, the transfer car is then transferred by the operator to the filling machine, where the vials are filled under aseptic conditions, and they are also partially stoppered under aseptic conditions. Once this is done, the filled and partially stoppered vials are transferred from the filling machine onto the transfer car by the operator and transferred to the lyophilization zone, where they are unloaded onto the lyophilizer. And the lyophilizer then removes the water content from the vial contents. And after this is done, the vials are fully stoppered. The lyophilized and fully stoppered vials are loaded from the lyophilizer onto the transfer car, and they are brought to the vial capping zone, where they are capped and crimped. This whole process can be either manual or automated. For automated, we don't have operator and transfer card. We just have a conveyor belt that transfers the unfilled vials from the vial depyrogenation stage onto the filling machine, and then partially stopping happens, and this is transferred by the conveyor belt back into the lyophilizer zone where the vials are lyophilized and fully stoppered, and these lyophilized and fully stoppered vials are transferred by the conveyor belt onto the vial capping zone where they're capped and crimped. So we have difference of manual and automated systems, where manual there's an operator, automated there's no operator. That's all. And once the vials are capped and crimped, they are inspected by an inspection device. Then they are labeled and packaged into a box and ready for distribution. And finally, we have the bioanalysis stage in the drug product manufacturing, where the vials, where the drug's purity, efficacy, and safety are confirmed. And bioanalysis is usually required for the following activities: backup of regulatory submissions, support of preclinical and clinical studies, monitoring of environmental conditions during manufacturing, and monitoring of quality during the manufacturing process. Yeah, that's my answer.
And that is a very good answer, Vignesh. That was very clear from start to finish. Well done. And the third question we'd like to get your opinion on is clean utility. So for drug substance and drug product manufacturing for a typical biopharmaceutical therapeutic, what would the requirements be for clean utilities?
Well, the requirements for clean utilities for typical, sorry, can you hear me?
I can hear you perfectly. Yes, indeed. Thank you.
Okay, right. So for clean utilities for typical biopharmaceutical manufacturing facility for drug substance and drug product, there will be 4 clean utilities. They are major clean utilities, and they are purified water, water for injection, clean and pure steam, and clean in place systems. That's my answer.
Excellent answer. I'm now going to stop that recording. Thank you very much, Vignesh.